Hello again. I can see that many people are already in the audience, so I guess we can go ahead and start with today's webinar. My name is Burak Ruperez. I will be the host and the moderator for this session. As you know, today we are going to be talking about the genetic potentials of the broiler chicken to optimal performance. We have very valuable speakers that are going to be with us, and they are going to share their know-how and their experience. Dr. Richard Bailey, from Head of Applied Physiology in Aviagen, and also Jose Maria Ros Felip, our Global Technical Manager from Perstorp Animal Nutrition. In today's agenda, I will be shortly giving you a small introduction about Perstorp. Then we are going to be listening to the presentation from Dr. Richard Bailey for about 40 minutes, and at the very end, Jose Maria Ros Felip will be sharing his opinions and his presentation with us, and then we are going to have a short questions and answers sessions. So please don't hesitate to ask any type of questions across the webinar. As you can see on the dashboard of the GoToWebinar, you are going to see a small arrow. Your microphones are automatically muted. This is not going to be able to turn them on. If you want to ask a question, please click on the orange arrow, and then you are going to see a chat box where you can actually put your questions there. This webinar is being recorded, and after the webinar is finished, we are going to make an available link for the rewatch, and also you are going to see again on the dashboard of the GoToWebinar that there's a small hand, there's a handout. You can download our Gastrifix Avi brochure in the hands-out section. At the end of this webinar, your support is, of course, as always, is highly appreciated. We are going to have a survey, and we are going to ask your opinion. We are going to ask your opinion about your feedback and how did you find the slides and how the webinar went. And of course, it's just a very short survey. Just feel free to fill those questions as well. Now, just a short introduction about Perstorp in brief. We have been working with short-chain fatty acids for a very, very long time. We have about 141 years of professional expertise, and we have different production sites across the world. In 8 different production sites, we are managing these main molecules that we are working together with it. And we exist in more than 60 countries with our Perstorp animal nutrition products. As many of you might already know that we are the major producer of the propionic acid, formic acid, butyric acid, and valeric acid with different applications, such as, for example, in propionic acid, we have our Prosid MI products, which are dedicated for preservation of the nutritional value of the raw materials and also to reduce the mycotoxin risks related with that. We also have very good acidifiers and also feed hygiene products to ensure the quality of the feed, to acidify the feed, and also to protect the animals that way as well, and to support them as well. And also, as you may know, that we deal very, very heavily with butyric acid and valeric acid, which are the 2 very important molecules that are essential to the animal in means of gut health. And then we also have our product range dedicated to these molecules as well, and our constant research and development is ongoing. And as you can see here also that the research and development is a very important heritage for Perstorp. Since, as I said, since 141 years, we have started our journeys first with the acetic acid, then also in 1990s we started the propionic acid production. Then we have actually purchased the Vaspic site that we are producing most of our animal nutrition products there now. And in 2012, we are the first ones to launch the esterified butyric acid, which is our Proforce SR. And just in 5 years, we went ahead with the Proforce Valerins also, which is the esterified valeric acids. And later on, we just kept on working on our research and development. And as you know, that also this year, for example, we have been launching a new product. This is just a peek to our heritage, and we are hoping that we are going to be bringing more value continuously with our research and development as well. Said that, soon I will be passing the word to our main speaker, which is Dr. Richard Bailey. He is the head of Applied Physiology in Aviagen. Many of you already recognize him, and many of you already know him. He's a very well-known speaker on the subject of gut health, especially, and the role of genetics. He has degrees in veterinary science and animal genetics and a Ph.D. in microbiology. And he has been working for Aviagen already over 12 years in the research and development department, in the R&D department, focusing on poultry health research initiatives. And he will be today sharing his opinions about the gut health and the breeder's perspective on that. So I will be making him a presenter now so that he can share his screen. Please, Richard, go ahead. And welcome, and it's very nice to see you again.
Thank you. I haven't got the option to share.
Please try now.
There we go.
We can see your presentation full screen.
Oh, you can. Okay.
Yes. Thank you very much.
Thank you. Yeah, thank you for the invitation to talk today. It's my pleasure to be presenting today on the topic of gut health and how we see it from the breeder's perspective. So in terms of what we do at Aviagen and within the breeding program, we have the elite stock, the pedigree stock, where we make our genetic improvements. And from there, the new genetics go down the different layers of the breeding pyramid through GGP, GP, PS, broilers, until finally we get to the consumer. Now, one of the interesting things, and something we always have to be thinking about, is that anything we do in the breeding program today takes 4 years to get down to the broiler level. And so we always have to be thinking ahead in terms of our directions of selection and trying to incorporate everything that we think the market is going to need for the coming years. And when it comes to selection, over the years, there has been an evolution of the selection criteria. In the 1960s, selection was basically based on the heaviest broiler and then the breeder which produced the most eggs. So 2 major traits. But nowadays, we evaluate over 40 different traits covering both broiler and breeder performance. But very importantly, we have within our selection criteria measures of welfare. And this can be skeletal integrity, cardiovascular fitness, immune response. And so we look at all of these kinds of traits, in addition to your standard broiler traits like meat quality, breast yield, body weight, and then the breeders in terms of egg number, hatchability, etc. And so having this wide range of selection criteria, we can do balanced breeding where we improve all the performance characteristics that we want, but we maintain that good, strong welfare aspect to make sure that the bird is healthy. And when it comes to decision making for selection, as I say, we're obviously interested in improving the broiler and breeder performance traits, but we also have to base our decisions on what is happening in the world. And this can be sort of aspects of welfare, new regulations, consumer perceptions. We had to think about antibiotic-free production, disease challenges, whether these are diseases that are new diseases or sort of older diseases that have sort of re-emerged. We also look at advances in science. How can we take new technology into the breeding program to make sure we're getting as accurate selection of the birds as possible and finding new traits? And so our breeding strategy is always evolving to make sure that we can do the best for the bird. And When it comes to the selection of the bird, we're looking at genetic potential. And genetic potential of the broiler relates to its innate ability to express a trait. And if we think of bird performance, these traits can be livability, robustness, body weight, breast yield, biological efficiency, just as a few examples. But the expression of these traits depends upon the genetics of the bird, what genetics have been passed from the parents to the offspring to drive the expression of these traits. But importantly, we also have the non-genetic factors. And these are environmental factors such as management, housing, diet, the health of the bird, gut function, immune function, and all of these things interact with the genetics to get that final performance of the bird and get the expression of that genetic potential. And understanding those interactions is one of the key things that we do to make sure that we understand what's happening inside the bird. And the influence of genetics to get this genetic potential varies from trait to trait. So some traits are under the low influence of genetics. And we measure this in terms of heritability. And the heritability is the proportion of a trait under genetic control. So if we think of traits which have low heritability, these are very often traits related to disease and mortality, things like that, because there are a lot of factors that can determine whether a bird is going to succumb to a particular disease or whether it's going to actually get the particular issue. So these traits are very difficult to select for because of that low heritability. Conversely, we have traits which have a high heritability, where they're under a greater influence of the genetics of the bird. And an example of this would be things like body weight or breast yield, where we have a large proportion of that trait under the influence of the genetics of the bird. And managing sort of how we select traits really depends upon heritability. When we think about gut health and the immune function of the bird, we have to maximize broiler performance in a wide range of conditions. So here are a few pictures of farming applications around the world, and it can vary from having the sort of most high-tech, environmentally controlled broiler shed to more sort of simpler ways of growing chickens, where we have open-sided sheds, we have sort of no environmental control, litter quality might not be as good, feed quality might not be as good. And so we have to think about how we can select our bird in this wide range of environments. And so then that makes us think about gut health. And when we think about gut health in the life of a chicken, it is dynamic. And so it has to be a key focus as soon as the egg goes into the incubator, and then when the bird goes onto the farm. And understanding the relationship of incubation, early life, and then the final performance of the bird is critical. For example, if an egg gets too hot during incubation, then the development of the gut tissues can be impaired, which means we'll have a deficient gut in the chick, which could then impact upon the bird through its whole life. Similarly, if we have poor early growth of the bird, so poor brooding, that will impact upon the development of the gut early on, which will then impact upon broiler performance. And so when we think about the gut, and gut health, it's a relationship between the gut tissues, the gut immune system, the gut microbiota, the time, age of the bird, the feed that's going into the bird, and the environment. And so selecting for gut health is really a tricky area to look at because it is so complicated. And what can we do to optimize gut function and health? Well, for this, we need to understand nutrition, we need to understand the environment the birds go into, we need to understand the host genetics, tissue development, how the immune system works, how that all links in with performance parameters, what we can do to improve water quality, and also another whole factor when it comes to gut health is the microbiota and the bacteria that live inside the bird and how that can drive gut health. And so understanding the links between all these influencing factors is key for continuing improvement in gut robustness. So if we always think about the gut as being the organ for digestion, which of course it is, But the gut is also a major immune organ. It's one of the major interfaces between the bird's immune system and the outside world. Around 70% of a bird's circulating immune cells actually reside in the gut tissues. So we have to think about gut health in terms of the immunity of the bird. So for example, in this cartoon, we have the gut, where we have the layer of gut cells, which are held together by tight junctions, which stop the gut bacteria from passing into the gut tissues. And keeping this these tight junctions strong is very important to maintain gut integrity. But then we have the interaction of the gut immune cells with the bacteria. And so here we have two examples of immune cells. We have the dendritic cells, which kind of stick an arm through the tight junctions through the gut cells and sample the bacteria that are in the gut. And then we also have the M cells, which are another kind of immune cell, which take sort of contents of the gut. And both of these cells process what is present in the gut and learn what is helpful bacteria, what might be pathogenic, and basically educate the bird's immune system to know what is normal and abnormal. And this early interaction of the gut immune cells with the gut bacteria is critical in that early maturation of the gut tissues. And so we have to really think about gut health from the minute the birds go on the farm, because this whole process starts very early on. And if we don't get optimal interaction of the immune system with the gut microbiota, that can increase the risk of things like dysbacteriosis later in the life of the bird. If we now think about how the gut works, it is a finely tuned organ and it requires optimal balance to work correctly. So here in this cartoon, we can see that we have the intestinal contents, which will contain the digester, but also the large community of bacteria that reside in the gut. We then have a layer of intestinal mucus, which covers the villi. And the villi are very important because they increase the surface area for the absorption of nutrients, but they also do play a role in actual motility within the gut. And one thing that's very important is that the villi has a very good blood supply, because we want the nutrients to be, once they're absorbed, to be carried around the bird's body. But also because there is blood in the villi, that blood leaks oxygen into that mucus layer. So we end up with a slightly oxygenated environment within the mucus, which acts as a protective barrier against strict anaerobes, for example, like Clostridium perfringens, that cannot grow in the presence of oxygen. And what happens during the normal daily life in the gut is that we have the peristaltic movements of the musculature down the gut, which creates a wave-like motion down the gut, with the blood pumping inside the villi. The villi pulsate. So we end up with this mixing of the intestinal mucus with the intestinal contents, which brings the nutrients down to the gut surface for absorption. And maintaining this is very important, because if there's anything that increases the viscosity of the mucus layer, so for example, if the nutrition isn't correct, or if we have an infection with something like coccidiosis, which causes an increase in the amount of mucus, this whole mechanism is impacted. And suddenly we lose that mixing of the intestinal contents with the mucus, which means we no longer get those nutrients brought down to the gut surface, and we lose out on the absorption of nutrients. And the problem that this occurs is, firstly, there are more nutrients for the bacteria, so we get an overgrowth of bacteria. Also, certain bacteria really like to use mucus as an energy source, so they start to proliferate. And when this lack of mixing occurs, we start to lose oxygenation in that villi mucus layer, which means certain bacteria, which previously were inhibited by that oxygen, can then start to colonize. And this is what happened with necrotic enteritis, where suddenly the Clostridium perfringens is no longer inhibited by oxygen. It can then come down to the gut surface, form a biofilm, and invade the tissues and start to cause disease. So maintaining that proper gut function is really important for nutrient absorption, but also to inhibit disease. So then another question that we often get asked is, how do we select for resistance to pathogens? And can we select for resistance to enteric pathogens. And this question really depends upon how the target pathogen interacts with the host, what kind of receptors it uses, what kind of conditions in the host will allow it to infect. With disease, do we want resistance, complete resistance to the disease, or do we want resilience, where even in the face of infection, the bird is still able to grow? And what are the practicalities of a disease challenge in terms of selection? Because to select against a pathogen, you usually need to infect an animal. And so that can cause problems in terms of for the welfare of the bird and things like that. So there's all of these kinds of questions when it comes to selecting for resistance. We have looked at the resistance to important pathogens. For example, Campylobacter, which is very important food pathogen for humans, and coccidiosis, which we know is one of the major intestinal diseases in poultry. And we find that the resistance to these pathogens is generally very low in terms of heritability, which again makes it very difficult to select for, because it just shows that there's so many environmental factors that can drive these diseases in terms of the manifestation in the host. And in addition to that, many factors can influence whether a pathogen will colonize and how it will impact upon the host, because in some cases a pathogen can only cause disease if the host is immunosuppressed or something like that. So in terms of disease resistance, it's a very tricky thing to select against. And so in order to get around sort of that kind of sort of those issues, we use what we call multi-environment selection. And what this enables us to do is essentially use family data from birds in an optimal environment, where we have the selection candidates, where we can get their full genetic potential. They get good food quality, good water quality, the environment is optimal, very high biosecurity, low stocking densities. So the birds are really able to express that genetic potential. We get the full sort of selection for heart and lung function, leg strength, livability and growth, and this environment is antibiotic free, and so there is no assistance from any medication in these birds, and from that we get the best out of the birds. But obviously this does not represent real life farms, and so we have our other environment, which is a more commercial environment, which has been we've been selecting we've had since 2000. And so 22 years now. And in this environment, we get the brothers and sisters of the selection candidates. And here we have low litter quality, we have poor feed quality, the lighting isn't optimal, and I'm really just sort of representing the lower quartile of farms globally. And in this environment, we can test the bird's robustness, the gut and digestive function, immune function, because we use built-up litter, so we have all of that bacterial and viral load in that litter. And we can test the livability, the uniformity, and the growth. And in this environment, it is antibiotic free, and we don't use coccidiostats. So there's a huge amount of commercial-like pressure in this farm. And then from this, we can look at the genetic link between these two environments, and we can look at which families perform well in both environments. And by using a commercial-like environment with built-up litter, we have lots of different kinds of microbial challenge, and very relevant microbial challenge to the commercial broiler production. And so it gives us a more holistic way of selecting for resistance or resilience to disease. And it really comes in with the part of that balanced breeding of being able to look at birds in terms of full genetic potential, and then those birds that are under a little bit more pressure. And in both of these environments, we can measure feed efficiency. So we have FCR measures in the pedigree environment, that optimal environment, and then we have FCR cages in that challenging environment. So we can get real life understanding of what the FCR is in those environments, and that will then link into what the birds experience out in the field, just again to get that balanced breeding. And if we look at what these two environments look like, in terms of performance, here we have mean body weight at 35 days of a broiler in both environments. And what we can see is that there's around 700 grams difference in body weight between these two environments due to poorer feed quality and that gut and immune challenge that we see in that commercial environment. So what we essentially achieve is through the optimal environment, we maximize genetic potential. And in this more commercial-like environment, we maximize robustness in terms of gut robustness and immune capabilities. And one thing we always make sure we do is check that we're getting relevant challenges. And by one thing we really do is look at the intestinal health of the birds in these environments. So for example, here on the left, we see some good gut tissues and good villi development in the optimal environment. And then what we see in the commercial environment is intestinal health issues. So mucus buildup, loss of the gut tone, watery gut contents, inflammation, damage to the intestinal villi. And this just ensures that we're seeing the kinds of problems inside the birds that we see out in the field when we look at commercial broiler production. Another thing that we check on as well is what is happening in the microbiota. And the gut microbiota is a key feature of gut function and gut health. And our investigations have shown that there is a shift in microbiota when you have good gut health and bad gut health. And these kinds of changes are also seen in birds in the field, which means that we have a relevant challenge in our commercial challenge environment and in our optimal environment. So on the chart here, this is sequencing of bacterial communities from the cecal communities in birds with good gut health and poor gut health, and then relating that to field cases where we have seen good gut health and poor gut health. And what we can see is when we have good gut health, we have more bacteria related to good fermentation and the production of organic acids and short-chain fatty acids like with bacteria like Ruminococcus, Butyriccoccus, and Allostipes. And we see them both in our internal environment and out in the field. When we start to see poor gut health, we see a shift in the levels of bacteria relating to good fermentation, and we see bacteria that are related to more malabsorption of nutrients from the small intestine. For example, with Odoribacter, Holdermania, we see more E. coli being present, showing that we have a challenge in the gut which is relating to poor gut health. And again, this is just a way that we make sure that the challenges that we have within our challenge environment are relevant to what we see out in the field. And one thing that we can also look at is also what happens with nutrient absorption. So we have a few measures that we can look at the bird. We can take a sample from the bird and assess how well it is absorbing nutrients, and we can relate that to gut score. And so with gut scoring, there's a lot of different techniques out there with different companies having different approaches to gut scoring. But essentially, it's where you assess gut tissues for inflammation, watery gut contents, gut tone, presence of coccidiosis, and that kind of thing. And what we see is that as we go from having good gut health, so very low gut scores, where we have good nutrient absorption, what we then, as that gut score increases, that we see a reduction in nutrient absorption. And when it comes to what we see in our pedigree environment, that optimal environment where we see better gut scores, so those low gut scores, we see more nutrient absorption. In the challenge environment, we start to see a loss of nutrient absorption relating to challenge. So through selection, we do have a long-term trend in terms of biological efficiency. This is data from the US, and we can see from 1925 all the way down to 2015, when this particular 2016, when this particular graph was published. There's been a huge reduction in FCR, and with that, we've also seen a huge reduction in mortality. So we see that the bird is becoming more efficient, using on average 38 grams less feed per year, and seeing a reduction in mortality of about 0.14%. And so through selection, we do see an improvement. And there's been some work done by Zuidhoff et al. who showed through 48 years of improvement of genetic selection that we have seen a big increase in the body weight of the birds, around 4 to 5 times, an FCR reduction over that time period of 50%, with an 82% increase in breast yield. And so from this, it just highlights what we can do with genetic selection by improving performance of the bird, but noting that we do see a reduction in mortality as well. And because we do that balanced breeding where we look for performance of the bird, but we do need to make sure that we select for that robustness in the bird as well. And in a world of increasing demand for food and competition for resource, having good growth in FCR is becoming more and more critical. And one of the things that we often ask ourselves is, how has this improvement in FCR happened? Is it that the birds are just more able to put the nutrients that they absorb for muscle growth? Are they getting better at absorbing nutrients? And this is some work that was done by a student in the US, Massini, who in 2012 did a project where he looked at the intestinal function and intestinal morphology of a heritage line versus a modern broiler line. And at 42 days of age, he found that when you look at villi length, villi width, and then the overall surface area of the intestinal tract, that the modern chicken has around 55% more surface area in the intestinal tract. So through selection for performance, for FCR, we have resulted in a broiler which has a gut that has more opportunity to absorb all those nutrients from the gut. And so that is really important, and it also highlights how important gut health is, because we've got a bird which has this huge capacity for the absorption of nutrients. So we need to make sure that that we support that gut tissue throughout the life of the broiler. Another thing that plays a major role in gut health is water utilization. And because we know that if the intestinal tract gets too wet, it can impact on nutrient absorption. It can result in wetter droppings going into the litter, which can then cause wet litter or foot pad dermatitis. And so we have looked at water utilization in the birds. And also water efficiency is important because we don't want the birds to be drinking too much water if they don't need to, because water is a precious resource. So we have to make sure that birds have good feed efficiency, but also good water efficiency. And something we found that is in the literature and we've investigated is that the avian kidney is quite different to that in mammals, and that it actually has multiple types of nephrons. And the nephrons are important because they are the structures within the kidney which filter all the waste toxins from the blood, and then we create urine. In the mammal, we're able to filter the blood, and the urine that then gets passed and out goes through various processes to reabsorb water. So mammalian kidneys can produce quite concentrated urine, and they are quite large. Birds also have a reptilian type nephron, and within these nephrons, they do not have the ability to concentrate urine. There is no water reabsorption from these nephrons. And these nephrons are quite a little bit smaller than that of the mammalian type. And what we find is that different birds have different ratios of mammalian to reptilian type. And generally what we see is that those birds which drink more water tend to have more of the reptilian type. And those birds which are more efficient with water have more of the mammalian type, which means they're actually able to concentrate their urine and reserve water, take water back so they don't need to drink as much. And so we've thought about water intake and what that role plays in gut health. And so we are actually able to identify those birds which have a higher than average water intake and use that information to improve water efficiency. And we can do that in the chicken, and we also do it we have a turkey breeding operation. We also do that in turkeys. And so we can make sure we breed an animal that is efficient when it comes to water utilization. And in terms of what sort of birds we would select when it comes to water intake, here is a graph of individual birds. Each dot represents one bird, And we have feed intake on the horizontal axis, and then we have water intake on the vertical axis. And if we take, for example, this point here, where we have a feed intake of around 1,300 grams, we can see that there's a wide range of water intakes. And when it comes to which birds would be selected, we would go for the average birds. We wouldn't go for the birds that really were underneath that trend line, because we don't want to be have birds that don't drink enough water, because obviously it's such an important metabolic nutrient. But then what we do is we select the birds which drink excessive water, water that they don't need for metabolic processes, or it could be that they play with the water stations, so they waste water that way. And from that, we can just make sure we select a bird which has optimal feed intake and optimal water intake. And in terms of the impact of this kind of selection, there's some work that was done by a well-established scientist, Rick Van Emos, and he showed that over the last 15 years of selection of broilers within our environment, that the modern broiler uses half a kilogram less feed to get to 2.5 kilos. And with that, we also see that it uses around 1 liter less water. And so when we multiply that up over the millions and millions of broilers that get grown globally on a yearly basis, that is a huge amount of feed and water that is saved in terms of resource. And saving the resource is really important, because if we look at the future sort of prospects of competition for resource, as we have the growing population, where it's estimated that the population will reach 9 billion people by the year 2050, the amount of arable land per head is going to go down to around 0.2 hectares of land per capita, compared to 100 years before to 2050, where there's about 1.5 hectares per capita. So as the population is increasing and the demand for food is increasing, the land available to actually produce cereal crops is decreasing. So where we have a broiler that has that ability to grow well with less feed input, that is really important when we look at this future prediction. And the same goes for water. As the population increases, there's going to be a greater demand for water for humans, obviously. And so we need, if we have a broiler which is able to use less water, then that is really important as well. When we think of feed efficiency, it is, as I mentioned previously, a key contributor to sustainable production. And when we look to the future within our own breeding program, we can see that we predict a reduction in FCR, and that reduction is going to improve as we go into the next few years. And we continue to do this, so we continually see this improvement in biological efficiency in the broiler. And then another thing that is also really important is becoming more and more of a focus around the world is the carbon footprint of agriculture. And so one thing that we can look at, if we look at the carbon footprint of broiler production, going from back in 1972 and then going forward over the next 10 years or so, we can see that the CO2 equivalent is going down. So with our improvements in FCR and the reduction in feeding input into the farm, reduction in water intake into the farm, we can see that we see a 1% yearly reduction in carbon footprint of broiler production. And this is a really critical figure, because it just indicates that broiler production is incredibly sustainable when we compare it to other animal protein production, and also when we think of cereal crop production and plant production. You know, we have to take all this into account. So looking to the future, and we have to think about emerging trends. And one of the big things we have to think about is consumer preference and consumer knowledge. As people get more well informed with things like the internet, social media, people are becoming more aware of what goes into food. So we have our traditional drivers, which are things like price and convenience and taste, where really the economic efficiency comes into play. And this is things like FCR, weight, yield, livability in chicks. So we're getting the most out of our birds to make the product as affordable to the consumer as possible. But then we have the evolving drivers, and these are the ones that really people are becoming more aware of. And so this can be the health and wellness of the birds, where people are concerned about the use of antimicrobials and other drugs used for therapy. And so this is where the livability, robustness, and gut health really come into play. And our multi-environment selection will really help select for this robustness and gut health. We also have sustainability and welfare. This is becoming so important globally. And as I mentioned, with our improvements for efficiency, we get that improvements for sustainability. But for welfare, that again is where robustness plays a role, and ensuring that we have a bird that is able to grow without any need for therapeutics and without showing any issues. That means that we have good welfare of the birds. But then also the skeletal support, metabolic support, and livability is very important, which we get from both our optimal and challenge environments. And so we have to continue to think ahead when it comes to consumer preference, and we remain to do so. So just to finish, a key thing with gut health is this breeding for sustainable production along with welfare. And gut health and gut robustness is critical for feed efficiency. And genetic improvements we can use to bridge that gap between global population growth and agricultural resource, and by minimizing the environmental impact of poultry production. Our genetic improvements allow us to capture those things. And we have a breeding goal to address that balance between biological efficiency and welfare. So by pushing both the performance traits and all these welfare traits that we do, we can make sure that the broiler goes in the right direction for the market. And we also have a very broad gene pool. So we have a huge range of pure genetic lines, which enable us to cover all market requirements and all specifications, so that we can ensure optimal performance in gut health and biological efficiency in all markets. And with that, thank you for listening, and I think we'll have some questions.
Thank you very much, Dr. Bailey. That was, as always, very interesting subjects. There are some questions already, which we are going to save it for the final part, for the questions and answers parts. Now, our next speaker is Jose Maria Ros. It's our global technical manager in Perstorp Animal Nutrition. He has more than 35 years of experience in animal health and nutrition by working in technical and marketing areas for companies dealing in the field of animal health, premix, and feed additives. He is also a member of the Perstorp steering group for development of new molecules based in the organic acids esterification technology. So with that, I'm going to give the opportunity to share his screen. And welcome, Jose Maria. We are looking forward for your chat.
Thank you, Burak. Can you see my screen?
We can see, but it's in the notes mode. Can you swap the presenter's view? Yeah. Thank you. Now it's working.
That's how it is. Okay. Thank you, Burak, and thank you, Dr. Bailey, for your very interesting talk. I think it's quite impressive to see how complex is the genetic selection is in the poultry production, is becoming in the poultry production. And I would like to highlight 2 points that can help us to introduce the topic today. Our topic today is how the relevance of the gut health and the robustness of the digestive system plays a role in getting the best of the poultry production. Well, we have just a few minutes. I just would like to highlight and guide you through what's possible, or the roles that the short-chain fatty acids can play around this topic. And in order to do so, we will very briefly touch upon these topics that you can see now in the screen. So the types of short-chain fatty acids and their production, how do they perform towards gut health? We will focus on the most studied one in that direction, in that field, which is the butyric acid. Some hints about the new one, the valeric acid, which is the longest short-chain fatty acid. And why do we feel that can provide with a good new tools to gut health, and how the combination of the two is behaving towards the improvement the performance of broilers. So, short-chain fatty acids, I think it's a very quite familiar term to all of us, and we all associate it to 4 molecules mostly: formic, acetic, propionic, butyric. All these are naturally produced in the gut by the fermentation of the non-digestible carbohydrates by the gut microbiota. There can also be the fermentation result of the fermentation of amino acids or proteins, but that's just a side amount of it. But it's not only about these 4. Actually, there are 8 that can be found naturally in the gut. And this is about the valeric acid, which is the longest of them. It's the C5. And there are other 3 that are just isomers of these molecules. But anyway, all of them are present naturally in the gut, and we can also assume that, like the other 4, the 4 well-known, they can also exert positive roles in the gut. In general, the role of the short-chain fatty acids has been studied in depth over the years, and this is just quite a recent drawing that has been published in the 20s, summarizing all the effects of these short-chain fatty acids towards gut health. So, and we can summarize it here: the epithelial function, which is quite well known. It's short-chain fatty acids are primary sources for energy to the enterocytes, but they also are active molecules in the transport of electrolytes, of nutrients, minerals, calcium, for instance, and the pH regulation. Along with this, they can also influence the production of the quantity and the quality of the mucus. They activate the globet cells in the intestinal lining, in the mucosa, to produce this substance, which is an active barrier defense and also relevant for the digestive process, just as we've been listening from Dr. Bailey. Barrier function, also well known and easy to understand the relevance for that, because if we do not have a full, clean and up to work intestinal barrier function, we can have problems with the absorption of nutrients and the pathogen passage into the blood. So they can support integrity, but also it's well known that they, especially butyric acid, they strengthen the tight junctions, preventing the excess permeability in that mucosa. Effects on the antimicrobial peptides, they can modulate their production. So as they can modulate the immune system. It's well also documented that short-chain fatty acids can reduce the production of cytokines, inflammatory cytokines, reduce the oxidative stress, but also upregulate the production of immunoglobulins. Outside of the physical structure of the intestinal lining, they can have also interactions in other body systems, but for instance, a nervous system, where by interacting with it, they can modulate the motility of the gut, so the passage of the nutrients all along the digestive tract. Well, as already said, butyric acid is the most studied one, is the best known and the most used with regard to gut health inside the intestines, I mean for activities inside the intestine. We've just chosen this table on the right, which is also taken from a quite recent publication, just summarizing a kind of meta-analysis, the type of effects that has been proven for this molecule. Doesn't matter the source, whether there is free butyric acid, coated butyric acid, esterified butyric acid, we can always expect positive results as the ones that we've seen in the previous slide. Positive effect on transporters and in the production of catechidins or the immune modulation, the villi length, and so on and so on. But what about valeric acid? Well, it's as I said, it's the longest short-chain fatty acid. That means that it's naturally produced in the gut, it's naturally present in the gut, and it makes sense to expect some positive outcomes, just as their brothers are doing there, no? The brother molecules. And there's little information about it, but there are very good indications for in human health or also in lab animals that it plays a positive role. And you can see in the drawing in the right side has been related or associated to valeric acid: a better recovery of body weight gain after sickness, prevention of inflammation, maintenance of gut integrity, stabilization of the gut microbiota, which is something that sounds familiar to us because we've seen it already for butyric acid. But it's interesting to note that the amount of valeric acid in the stools is also associated with the level of health of the person. So the more valeric acid, the healthier people. Digging a bit deeper in the mode of action, it's been also noted that it can inhibit the HDAC, so the histone deacetylase enzyme, which is associated to pathogenic processes such as colitis or cancer. But that's in humans and lab animals. So having that in mind, we decided to run a project with the University of Ghent, a 4-year project. We're trying to learn more about this molecule. And we immediately noticed that the effect on the villi length and the grip depth is quite clear, just as we've seen for butyric acid, but they can also have a positive impact in the L cells. The L cells in the intestinal mucosa are the ones that can release or produce glucagon lactopeptides signaling molecules, GLP-1, GLP-2. And these molecules has also a systematic impact, positive for most cases, but especially in focusing on the gut health. We can highlight the effect on the intestinal mucosa development, villi length, as we've seen in the right side, in the left side of the drawing, but also the nutrient uptake, the control, the modulation of the inflammation, and many others. So quite an interesting effect for valeric acid. That was found in healthy birds, but also in challenged birds, we've seen positive results when valerians are in the diet. This is the result of a challenge trial run in the US, where we had a trial set with 4 groups, negative control, a positive control, and the other 2 groups challenge just as a positive control, but with the addition of bacitracin or monovalerians in the diet. I think it can be easily noted if you look into the performance and lesion score, performance figures and lesion scores, that both treatments, bacitracin and monovalerians, managed to improve in a significant manner the results of the positive control. Meaning that for sure bacitracin is doing a good job, but also monovalerin is also meeting the effects of the bacitracin. So also positive effects after challenge. Well, but then we have 2 molecules, butyric acid and valeric acid. We have a good experience with the butyric acid, positive for many years. After 10 years or 11 years of experience, we've noticed that and we can assess that. But we decided to compare the effects of these 2 molecules together with 2 groups, one a control group with no treatments and another group with tributylin in the diet. And this compared to a combination of butyrins and valerins, just trying to mimic the real environment in the gut. And we noticed that the tributylin outperformed the control group as expected, despite the control group was already doing a very good job, so 2 grams of daily gain and 1.55 feed conversion, so good results to meet or to outperform. But the group with the combined use of butyrins and valerins did even better. So there seems to be good promising results for synergies between the butyric and valeric acid. And that has been noted in the number of trials that we run later. It's about 8 trials. In all of them, we've been very consistent results as feed conversion improvement. 6 out of the 8 did significant improvements. The average performance is 4 points better as feed conversion, with the impact that will have in saving in costs, but also the body weight was also very good, positively improved in these animals. I would like to highlight that these trials, none of these trials were done with challenges, so in case of field conditions, we should expect even better results. In summary, the short-chain fatty acids can play a critical role to improve gut health through a number of different modes of actions. Butyric acid is well studied, is the reference for that, and we have a long experience with their use in poultry. Valeric acid is a promising new molecule, and the combination of these two are delivering very good and promising results to the future in the production of broilers. And with that, I can conclude my talk. Thank you.
Thank you very much, Jose Maria. That was very valuable information as well. Thank you for sharing that with us. Let me put this. Can you please confirm if you're seeing my full screen?
Yep.
Okay. Richard, if you can also turn on your camera, thank you very much. We have some questions. For the sake of the time, we will try to address as much as possible of those questions. The first one is coming to Dr. Richard Bailey. One person from the audience is asking, how do you do the gut scoring, and can you share some information on that, like if there is a specific approach that you have on gut scoring, gut health scoring?
Yep. So I use a technique that I sort of put together myself within Aviagen, and within that I try to keep it as simple as possible. You can get very complicated with the way that you assess gut health, and it can become then quite variable on how one person scores something versus another person. So I try and keep it simple in that I will look at, if we take a small intestine, I look for 3 key things. The gut tone, so when you cut into the tissues, how that gut opens up, any inflammation, and what the contents look like, whether they're watery or normal. And I'll do that for the duodenum, jejunum, and ileum, and score it on a 0, 1, 2. 0 normal, 1 mildly abnormal, 2 severely abnormal. Then look at the ceca, look at the color, look at the consistency, and again score that 0, 1, 2, normal, mildly abnormal, severely abnormal. Look at the gizzard, if there's any signs of erosions, again 0, 1, 2 in terms of severity. And then look for coccidiosis, as you know, the normal coccidiosis scoring. And then essentially you add up all those scores. And so the higher the score, the worse the gut health is. And the reason, like I say, to keep it simple is that I can give this scoring system to our technical guys, our veterinarians. And so when they come to me with scores, I have a good idea of what they're looking at. Because if you take something where you might have a score of 10, 1 to 10, my 5 might be someone's 3. And so it can then get quite subjective. So I always just try and keep it as simple as possible. That's how I approach it. The technique is actually published in a paper that we had published a few years ago on Campylobacter, where we use gut health scoring. That's in Poultry Science. And so if you search for my name and Campylobacter colonization, you'll be able to find the scoring.
Okay, thank you very much for that answer. The next question is for Jose Maria. It's a two-sided question, but I think it's also a good question. Can we use butyric acid for human gut health also? And the second part is that how to make it palatable because it has a very bad smell. So what would you say about that, Jose Maria?
Well, actually, butyric acid is used in human health. So And the evolution of the use of butyric acid has been following the same steps that we have noted in animal production. So butyric acid is butyric acid for humans, but also for animals. So it's not a nice molecule to work with. And over the years, the industry have learned to produce less nasty solutions. So from the free butyric acid salts coated, now the trend is clearly towards the esterified products. So we can say now, confidently say that the problem of the smell of butyric acid is not there anymore.
Okay. Thank you very much. It's already 11 o'clock here. We are at the end of our session. There are some more questions, but we are going to address them. We are going to, of course, share all of the answers from all of the speakers as well. I would like to thank to both of you very, very much for being with us here and then sharing all this valuable information with us. And we would like to thank to the audience also that they have been very, very attentive across the webinar, and many people have been watching this webinar. So we are very, very thanking to them as well for being here with us. And with that note, and if I don't see any of you, so to many of you, I would like to wish a Merry Christmas and a Happy New Year as well. And once again, thank you very much for being with us.
Thank you.